Diana, she's a postdoc now with Christoph Juncker in Vienna. And she did her studies in Romania. She did her bachelor in the Universitatea Polytechnică from Bucharest, where she did medical engineering. Then she moved to King's College in UK, in London, where she did her bachelor, not the bachelor, the master. And then she did her PhD there, where she mainly focused on neuroimaging, spectroscopy, mainly, if I understood correctly, GABA editing in humans, animals, mainly brain. And now she will be presenting us pancreatic MRS, proton MRS. And I'm looking forward for your talk. And yeah, something important, you probably, some of you know, but some of you do not know. The three of us, me, Diana, and Jamie, we were working on the multi-center preclinical MRS study. So in fact, we also gathered here to work a little bit on that. So yeah, I just wanted to thank you for being here again.
Thank you, Christina, and thank you everyone for joining my presentation. As Christina nicely introduced me, I've been in Christoph Juncker labs, and both at Columbia and now in Vienna. And as you can see, this project is a joint effort between two labs at Columbia, the Kenneth Olive Lab and the Christoph Juncker. Now, by me, a bit of introduction to add more on what Christina mentioned. As she highlighted, I did my bachelor degree at University of Vienna in Bucharest. Then my interest for spectroscopy and research started during an internship in Lyon, where I worked on near-infrared spectroscopy. I carried on my studies with a master's and a PhD at King's College London, both in neuroimaging and with the same supervisor. Then I moved on to my first postdoc adventure in Cambridge, where I worked on multiparametric mapping and hippocampus MRS. Then I moved to Christoph's lab in Columbia in New York, and that's where I started to work under an industrial collaboration as well, and dealing with more clinical studies. And now in the end, the third postdoc at Vienna, where I'm working on the pancreatic cancer study. Now, let me quickly introduce you to the pancreas and pancreatic cancer, and I will start with a brief middle school biology lesson on the pancreas. You might remember since years ago that it is an organ situated between the stomach and the duodenum. It has two major roles: an exocrine function that allows it to produce enzymes for digestion, and an endocrine function that allows it to produce hormones like insulin and glucagon for blood glucose regulation. When it comes to pancreatic cancer, of course, these functions are disrupted, which leads to impaired digestion and hormonal regulation. Worth mentioning as well, there are 4 stages related to pancreatic cancer. The first one where the tumor is confined only to the pancreas. The second stage where it spreads to the lymph nodes in the proximity of the pancreas. Stage 3, where the tumor reaches the blood levels, the blood vessels in the nearby region of the pancreas, and then finally it spreads to organs. Numerically, pancreatic cancer ranks 12 on the incidence scale and 6th on the mortality scale. It also has a very low 5-year survival rate of about 13%. Depending on years, you'll find it to be 5% or 18, 20%, but roughly around 13% over the years. These numbers actually make pancreatic cancer severely lethal. Now, there were several attempts, as you will see on the slide, of understanding the biochemical profile of the pancreas using proton MRS. And here are a few examples of papers that focus on normal and pancreatic cancer, with studies not only on humans, but also on animals, as you can see. However, if you take a closer look at the spectra, something might look a bit odd. Either the low SNR, the line-broadened peaks, the limited spectral information, or the prominent baseline, something in general doesn't feel quite right. In addition to these aspects, I would like to highlight that all these studies used the vendor-provided software, not a pipeline tailored to the pancreas. Of course, that comes with some disadvantages. Now, let's see what can go wrong and cause those problems. So, of course, we start with our subject, either human or animal. We want to look at the pancreas, so make sure we place the voxel as accurately as possible. We get our spectrum, and voila, we got out the chemicals. But it's not so easy, and we all know that. And the limitations and challenges that appear in pancreatic MRS start with the anatomical location of the pancreas, where you can see here that the pancreas wraps around the nearby organs, and also it's hard sometimes to distinguish it from the nearby anatomy. So we deal with issues related to size and shape, as well as location and proximity to air tissue interfaces, and of course the motion driven by the lungs, the heart, or the bowels. Then it carries on to the biochemical limitations, where here you have an example of pancreatic cancer models, where you can see here that the coding the choline peak here is resembling the normal choline one, whereas the phosphocholine and glycerolphosphocholine resembles another type of pancreatic cancer. So this leads us to the conclusion that there is a heterogeneous biochemical profile between different stages of pancreas and also different types of pancreatic cancer. Now, in addition to this aspect, we also have a limited knowledge, as you might remember from the reduced spectral information that we saw previously in the literature, at least with proton MRS. Then other limitations include the subject, human or animal, and it starts with their poor health state and the fact that the cancer actually progresses quite fast. So if you remember those 4 stages that I've showed at the beginning, sometimes it takes only 6 months to have that progression. So you need to move very fast and to be right on time to actually be able to get your spectra at the right moment. Now, in addition to this, when it comes to animal models, it's quite difficult as well to model the human pancreatic cancer to rodents. And you already saw that there are already 2 types of pancreatic cancer that are studied in animals. Now, the interesting part, the method limitations, and you probably are all aware of the consensus efforts and the publication of several papers that describe how MRS should be done, from terminology to data acquisition, processing, and analysis, and also to interpretation of the spectrum and the metabolites, as well as the macromolecules, as Christina highlighted in her paper. And what I notice here is that there is a lack of pancreas-tailored pipelines, mainly because the researchers didn't necessarily work with spectroscopists. They had some expertise available. They were under time limitations probably. So of course you need to deliver within that time frame with the best that you can get. Then this leads also to the preference for vendor-provided software, which are great, but they are not necessarily dedicated to that organ. So of course that comes at a cost of, a compromise in the end. And the lack of standardization and unapplied recommendations, that's also partly because these publications, all of these ones, at least for the papers I've shown earlier on, the consensus papers came out after those publications. So a bit late for those people to do something, but never too late. Now, the ones that I'm going to focus on are the highlighted ones in red, and these are the problems that we want to tackle, and the proposed solution is a pancreas-dedicated pipeline for spectral processing and analysis that aligns with the consensus recommendation and state-of-the-art MRS. Now, the general framework of the study actually starts with the mouse model of pancreatic ductal adenocarcinoma, which is the purpose of the research in the Kenneth Olive Lab, our collaborator. This study takes into account 2 groups of mice, an untreated group, a treated group as well, and obviously the effects of this treatment. The plan is to scan the animals at 2 time points at baseline, and then after the baseline scan, the treated group will receive, of course, the treatment, and then 10 days later, both groups are scanned. Now, in my presentation today, I will only discuss about the untreated group of mice that were scanned twice, and these data will be used to test the pipeline. Now, the pipeline that I keep talking about starts, of course, with the acquisition, where the animal preparation is very important, and aspects like anesthesia needs to be taken into account, as well as the careful placement of the voxel, as you've seen, because of the location and the shape of the pancreas. And then finally, we get our data. This data is fed into the processing pipeline, and we start with what you all know already probably, eddy current correction, frequency and phase alignment, and then quality assurance and removal of corrupted averages. This is actually a very important step, which is probably quite familiar to all of you, not necessarily so much used in all those publications that I showed earlier. Then only after these steps, we do the data averaging, and then apodization, zero filling, phase correction, frequency calibration, and finally we get so wanted process data. The steps highlighted in red are the ones that normally are missing from the pipelines provided by the MR software vendors. However, you'll see later on that this actually makes a huge difference between the data quality that we obtain in the end. Now, the second part of the pipeline includes the analysis. So we have the process spectrum that we worked so hard on, and we now want to understand, okay, what signals do we see there? And to do so, we created a basis set that we simulated with RF shapes and timings that match the data acquisition. We take these basis functions that give us the ideal spectral profile of each chemical we expect to see in the pancreas spectrum, and then we model it to the point that we get the fitted data. For the quantification, we take the modeled and fitted spectrum and we break it down into the major metabolites that we think will be there, and then we obtain the concentrations by calculating the peak integration. Now, more details about the data acquisition include the use of the Bruker 9.4 Tesla MR scanner with the sequence parameters that you can see on the slide. Most important thing is the use of a press sequence with a short TE and a rather large voxel size of 9 milliliter cube. That is an example of a voxel placement as well, and the corresponding process spectrum obtained with our software inspector, which is an in-house MATLAB-built software in Christoph's lab. Then the analysis is done with the same software inspector that allows the linear combination modeling of the basis functions. For the simulation of the basis functions, we use the software Mars, and this is an example of the output that we obtain. You all know already that we have the data shown in black, very similar to a C model, of course. The fit shown in red, and then the residual signal shown in gray, and we also have a residual water peak highlighted by the green band. Now let's move on to the important part, namely the compounds that we see in this spectrum. And I will start with the lipids that spread between 0.5 and 3 ppm, also beyond the water signal at 5.3 ppm. These chemicals resemble roughly the liver. However, the expectation generally in the field is that the lipids will be decreased in the pancreas compared to the liver. Also, these chemicals will change concentrations depending on the local condition, either healthy pancreas or cancerous pancreas. Then the metabolites, here you probably recognize some of them, like creatine, choline 1, taurine, glucose, glutathione as well. They usually spread between 3 and 4.5 ppm, and you can see here the linear combination of their contributions. These metabolites also are expected to change concentrations in the context of pancreatic cancer. However, it's still unclear how much will they change. It is known that they will be altered, but if it goes up or down, that's still to be figured out. And at the bottom as well, you can see that there are different combinations of metabolites that can be used when considering the pancreatic cancer. Now, in terms of concentrations, on the left side, you can see the metabolites, on the right side, the lipids. Then you have in green the distribution of concentrations at baseline scan, and then in purple, 10 days later at the second time point. There are no statistically significant differences between these two time points, which is exactly what we expected, given the fact that this is a short time window. There is not that much aging and not that much cancer progression happening during such a short time. So, of course, the concentrations will be quite similar between the two time points. And now in the discussion, let's talk about the deliverables. As mentioned earlier, the so wanted pancreas pipeline with the additional steps that include not only basic but also advanced preprocessing operations, namely spectral alignment. Also, this pipeline allows the addition of other modules like water removal modules with HSVD algorithm, and it also allows full control, but also responsibility of the user regarding the operations used and when and in which order are they used. Then the analysis is more sophisticated than the ones accessible with the MR software vendors, with the software provided by the MR vendors, namely because it allows the use of simulated basis functions that match the data acquisition and also the linear combination to disentangle the contribution of different metabolites. Then, like most other pipelines that are not provided by a certain vendor, it is vendor agnostic, so it will be compatible with any data formats. It's easily replicable and shareable given the nature of the software we use. and it's relevant to both human and animal data sets. Then, of course, very important aspect, it is finally aligned with experts' recommendations and with state-of-the-art MRS. Then the second outcome of this project was an updated biochemical profile of the pancreatic cancer. And you might remember from the earlier slides that usually there are 4 or 5 peaks that were identified in the pancreatic cancer in the normal pancreas. We managed to identify 9 lipids and 9 metabolites. But of course, there is still work to do in this regard. And despite our efforts, there might be other chemicals that we did not consider in our basis set and that will require further investigation. For instance, from the literature, when using other methods, not proton MRS like HR-MAS, as Jamie introduced earlier on, there were changes in lipids that decreased as well as creatine as observed in literature. There are also expectations regarding choline that it should increase in concentration, but there are papers either that use HR-MAS or another method that mention changes in lactate, alanine, valine, leucine, isoleucine as well, and also alterations that are not always seen by all researchers, but some of them do report changes in glutamate, glutamine, in myo-inositol, taurine, and glucose as well. Therefore, when it comes to understanding the biochemical profile of the pancreas, we should consider what happens in the cell during the evolution of the pancreatic cancer. This is a diagram that shows the vast immensity of reactions that happen there, and of course, the ones that I don't know yet, but now I need to step up my game when it comes to biology and chemistry. However, we do notice some chemicals that we are aware of, like glucose, we have lactate here, we have aspartate as well, we have leucine, isoleucine as well, glutamine as well, and BCAAs, these are usually valine, leucine, and isoleucine. So these all chemicals are involved in the progression of pancreatic cancer, and they do have a role, as shown there. Pancreatic cancer leads to this reprogramming of the metabolism of lipids, of amino acids as well, and glucose. So there might be changes that are observed in other chemicals than the ones that we included in the basis set so far. For instance, with HR-MAS, other research groups noticed changes in taurine, leucine, isoleucine as well between normal pancreas and pancreatic cancer. This is also supported by another study where they saw glycine and taurine changes. So obviously these chemicals might be visible in our spectrum. It's just that probably the contribution of the lipids is strong enough to hinder their quantification. But there is definitely an effect on lactate, because during the hypoxia, that's why I mentioned the hypoxia during Jamie's talk as well. During hypoxia, one way to use the glucose is to actually break it down and to produce lactate as well. So there's an expectation in increased lactate. Then the tumor cells will proliferate more than normally, so there is a need for the compounds to build these cells. So that's why we have increased lipid intake as well. That's why we need the choline as well, the BCAAs, which are valine, alanine, and leucine and isoleucine as well, will be needed. And then in addition to that, there is a use of energy that needs to be met, and it doesn't happen under normal mechanisms usually. So that's why all these chemicals will probably appear in the spectrum. It's just that in our case, we don't have the sensitivity of HR-MAS. So we might need to actually look at HR-MAS as well, and then combine and corroborate that with our proton MRS. And then, of course, all these efforts will go towards the study, the greater project that includes the data analysis and preprocessing of the treated group, and hopefully we'll discover a change between the 2 treated and untreated groups. And with this, thank you for your attention, and I'd like to thank the collaborators and third parties that contributed to this project.